Hello and welcome to our video notes for cell transportation vocabulary. Remember, this is a video, so play, pause, rewind, repeat as many times as you need to get the notes, to get the doodles, and to actually get an understanding of the content. Let's jump in. Today, we're talking about cell transportation and really talking about the movement of different types of molecules across the cell membrane. Before we get into the specific types of transport that we have, we first need to talk about why things move. So we're going to talk about this term, concentration gradient. So a concentration gradient is, that's not better, sorry guys. Okay, a concentration gradient is the gradual difference in concentration of a dissolved substance in a solution between an area of high density to an area of lower density. And that's a lot. So we're going to doodle. Really, what this means is that we have a solution of something. Let's draw a beaker. And it's got some water in it. Our concentration gradient means that there's an area with a lot of some type of particles and an area with not very many. And these particles will slowly move towards. So by the time we're done drawing, we should have a bunch of dots on our left side to show our high concentration, and just a few dots with a medium amount in the middle. So we have our high concentration and our low concentration. So this is why things move. If you're in class and it's super crowded in one area of the class, you're most likely not going to go sit with all of those other crowded people. Or like when we go to an assembly, you don't all cram into one space. You kind of like, oh, there's open space, and you move. Molecules are pretty much the same way. They don't want to stay where it's super crowded. They want to move to someplace that they have space. So, to be able to move, we've got two different types of transport that happen. We have active transport and passive transport. Let's start with active. So, active transport means transportation of materials that requires energy. So, just like when you're being active, you need energy. When molecules are moved through active transport, they also require energy. They require energy because they move against a concentration gradient. So instead of going from a high area to a low area, they go from low to high. So we could draw that with a few particles in a space. And let's draw a dividing swiggle. And then a lot more particle dots. And what active transport does is it uses energy to move these particles here are missing out on the party. They want to actually hang out with their friends. So they use energy to get up off the couch and travel to where all those other people are. Now, not every person likes to actually be around a lot of people, and not every molecule does. So not everything does active transport, but it is one way that cells move things. And active transport can be accomplished through a couple different means. So let's slide our paper on up. Our first is the use of a molecular pump. So molecular pumps are proteins in cell membranes that help them move molecules. So just like when we drew in our notes previously, we had a layer of phospholipids with their double tails and their single heads all lined up nicely. And going our phospholipid bilayer both directions. They don't have to line up perfect, it's okay. And then every once in a while we had a protein that went all the way through. And then our membrane continued with its phospholipids on its bilayer. So, if we're drawing this, we have our protein pump. This. It helps molecules move either into the cell or out of the cell with the use of ATP energy. Other types of active transport include endocytosis and exocytosis. And these are basically opposites of each other and work pretty simply like this. Let's start with exocytosis. So this is when materials move out of a cell. So just like exit is to go out of a space, maybe you exit your classroom or you exit your house, exocytosis is materials move out of a cell. So we could draw a cell, and let's give ourselves a little nucleus and DNA in there. And maybe there is some substance on the inside. In exocytosis, this moves out of the cell to now be out here. It takes energy to move the cell membrane and push that all the way out. So it's a type of active transport, exocytosis. The opposite is endocytosis, and endo, kind of like into, goes into a cell. So this time, instead of having our materials moving outward, we're going to have our cell with its nucleus, and maybe we have some material out here that the cell needs inside of it. So the cell is going to use active transport to suck this material in through the membrane, and endocytosis occurs and our cell moves in. So those are our different types of active transport. But cells need to move a lot of things and have a lot of other processes that also need energy. So they can't be using all of their energy just to transport things back and forth. So instead, they also have the use of passive transport. Now, passive transport is the transportation of materials that does not require energy. So for this one, things move with the concentration gradient. So they go from a high concentration to a low concentration. So we can do what we did for active transport. If we draw an area of a lot of particles, it's very crowded. Maybe all of these things feel like their neighbors are taking up too much space. And on this other side, we've got just a few particles. And maybe there's a barrier, like a cell membrane. In passive transport, we don't need any energy, but this material are going to be able to go the other direction from high concentration to low concentration to try and even out and reach a balance. And just like active transportation, passive transportation has a couple different processes in which things move. So, let's talk about those. Our first one that we're going to talk about is going to be diffusion, or simple diffusion. depending on which books you're reading and or who you're speaking with. Sometimes we make sure we specify simple diffusion because there is another type of diffusion. But diffusion, in the simple meaning, is the process where molecules move from an area of high concentration to an area of lower concentration. So basically, what we just drew. We have, let me straighten this out. We have an area of high concentration with lots and lots of particles that are all just bumping into each other and feeling crowded and generally would rather not be in this big globby mess. And we've got an area of low concentration and a divider of some kind, like a cell membrane. Simple diffusion means that these concentration of high simply just move to a low. Sometimes it's across a membrane, or sometimes it's just throughout a solution. Like if you ever drop a couple drops of food coloring into water, they start very concentrated in one spot and they slowly work their way out through simple diffusion. Our next type of passive transport is facilitated diffusion. So, facilitated diffusion, too far, is when large molecules need help from membrane proteins to move from high to lower concentration. So, if you facilitate something, you're helping it happen. In facilitated diffusion, it's still passive because we're moving those molecules from a high to low concentration. It doesn't take energy to go against the gradient concentration, but this time they're too big. They're not something small, such as an oxygen or carbon dioxide molecule that can just pass through. These are larger molecules like carbohydrates or amino acids or ions that have a charge. And so they need help. So this means that we have I'm going to draw my cell membrane a little bit differently this time. Cell membrane in its phospholipid bilayer. And in this still, we have protein, like a protein channel that helps large molecules move into or out of the cell. This is facilitated diffusion. So we have our cell membrane. We have large molecules. And we have a protein helping the movement happen. All right, our last one we're going to talk about is osmosis. And osmosis is really cool. So all of these other ones have been talking about the movements of molecules or proteins or carbohydrates in large things. Osmosis is kind of the opposite of those because it's the movement of water through a permeable membrane. So instead of the actual proteins or carbohydrates or other large molecules moving, it's actually the water that moves with osmosis. So to fit this in, we're going to draw 2 beakers. And each of our beakers should hopefully be relatively identical. They're going to have a selectively permeable membrane dividing them in half. in our first beaker, we're going to have a solution on each side that starts at the same level. One side has a relatively low concentration of particles, and the other side has a lot of particles. Now, in our other types of diffusion or active transport, these particles that we drew would move. But in osmosis, the water is going to move. So we start with a low concentration and a high concentration at the same levels. And after osmosis happens, we end up with something really cool. We end up with an equal or relatively equal concentration of particles, but our membrane now has different levels of liquid. So the water moved from the area of low concentration to the area of high concentration. So now these particles have room to space out. Basically, the water came in and made the room bigger instead of the particles having to move themselves. Kind of cool, right? All right, these are our notes for cell transportation vocabulary. Remember, if you didn't get it all, pause, rewind, go back, watch it again, turn on your captions, and make sure you get it all so you can use these words while we discuss and continue learning about cells and cell transportation. See you in the next video.